Hello learners, welcome to NR Studio. This is live eVidya program for Science and Technology Senior Secondary course of NIOS. I am Neelam Gupta, course coordinator of Environmental Science. Welcome you all this program. Learners, in our previous program we have discussed how life began and how such diverse forms of life that we see around that have evolved. Before we start discussion, we would like to take an example for making a wall. When we build a house, we build walls, then and you think a number of bricks are arranged end to end to prepare a wall. So now you have an idea, got an idea what I am talking about? Yes, I am talking about, trying to talk about our building blocks of life, that is cells. Like bricks in a wall, cells are also arranged variously to build the body of the living beings. Really, every organism begins their life as a single cell, which is as a fertilized egg. So cells divides to give more cells. Like when we build a house, while building house, we build first walls. So cells forms tissues, tissues makes organ makes organ systems, formation of a body. So during this program we will discuss about structure and function of the cells, how cells divide, how they collect to make a tissue, and how these cells are being used to repair damaged part. So we will talk cell division too, and some damaged parts are repaired by the cells through a technology known as stem cell technology. So what we are discussing this program, first we will talk about cell as a structural and functional unit of the all living organisms and the state of cell theory, different types of cells, that is prokaryotic and eukaryotic cells, what are the difference between a plant cells and animal cells. So we will talk about similarities and differences, cell organisms and the organelles, and say their functions. Why do we need cell division, or we can say importance of cell division, and we will talk about different types of cell division also, mitotic and meiotic cell division, tissues, and also describe various plant and animal tissues, and lastly we will share an idea about cell technology and stem cell technology and its use. So first, how cell discovered? Firstly, invention of microscope helped in the discovery of the cell. Robert Hooke, and English scientist, discovered the cell in 1665. Cell means compartment. So he observed a thin piece of cork under his simple microscope and saw many compartments arranged like a honeycomb. If you see honeycomb, so you can see how they arrange each cell. So cell is also similar as a honeycomb. So discovery of the cell. Why we say cell as a structure and functional unit of the living being? Because cell is the smallest complete unit of all living beings, and many cells form together tissue. When Robert Hooke and Leeuwenhoek discovered the cell, and they got established in theory that cell is the structure and functional unit of the organisms. So when Robert Hooke discovered cell, soon after discovering of the cell theory was formulated in 1839 by two German biologists, botanist Matthias Schleiden and German zoologist Theodor Schwann. They proposed a cell theory. According to this theory, cell is the structural and functional unit of the all living beings, and bodies of the all living organisms are composed by cells, and all new cells arise by division of pre-existing cells. The function of an organism are an outcome of the combined activities and the interaction of the cells that makes the organisms. So, when we talk about cell and how do we define, we already told cell is the structure and function unit of the living organisms which is capable of independent existence. So, now we will talk about different types of cell, but before types of cell we will talk about all cells have three basic parts. First, cell membrane which limits the cells and gives it shape, DNA which may be contained in a nucleus, and fluid called cytoplasm filling in the space within the cell. And protoplasm is the living substance of the cell, and nucleus and cytoplasm together form the protoplasm. So, when DNA of the cell lies in the cytoplasm or is enclosed within the nuclear membrane, cell may be termed as prokaryotic and eukaryotic. So, cells are of the two types, prokaryotic cell and eukaryotic cell. First, we will talk about prokaryotic cell. Pro means before and karyon means nucleus. These cells do not have well organized nucleus. We are talking about prokaryotic cells, genetic material in a single molecule of DNA lying in the cytoplasm. There is no nuclear membrane, and cell organelles like mitochondria, lysosomes, endoplasmic reticulum, chloroplast, nucleolus are also not present in a prokaryotic cell. An example of the prokaryotic cell is bacteria and blue green algae, because they are single cell organisms. Second type of cell is eukaryotic. Eu means true and karyon means nucleus. As name suggested, eukaryotic means nucleus present. DNA is enclosed in a nuclear membrane forming a nucleus, and membrane bound organelles like mitochondria, endoplasmic reticulum, Golgi bodies, lysosomes, chlorosomes, nucleus are present within the cytoplasm. And genetic material is made of two or more DNA molecules which is present as a network of chromatin fibers when the cell is not dividing. Examples: all plants and animal cell, fungi, protozoa, and including human cell. Now, we have talked about prokaryotic cell and eukaryotic cell. How can we differentiate? In three terms. First, genetic material lies in cytoplasm and genetic material contained in nucleus. Single chromosomes are present in prokaryotic cell. Two or more chromosomes are present in eukaryotic cell. Organelles are absent in organelles like mitochondria, Golgi body, chloroplast are absent. In case of eukaryotic cell, organelles are present in cytoplasm. So, now we will talk about structure of a typical eukaryotic cell, structure of a typical cell. Cell with eukaryotic means Eu means true, karyon means nucleus, and all organelles are found in eukaryotic cell. So, cells within the body of a multicellular organism differ in shape, size and function, but all have three basic parts, that is cell membrane, cytoplasm and nucleus. So, both when we are talking about animal cell or plant cell, both have plasma membrane or cell membrane, cytoplasm and nucleus, but some parts are absent in plant cell or some are present in animal cell. So, first we will talk about the common parts, common cell organelles. First we will talk about plasma membrane. Plasma membrane is the delimiting selectively permeable membrane of all cells and regulates entry and exit of the material from cell. A thin delicate membrane enclosing the cell, and plasma membrane forms outermost covering in animal cell and the inner to cell wall in present plant cell. They are selectively permeable, and functions of the plasma membrane is selectively permeable so allows only selected substances to pass in and out of the cell, protects cell from injuries, and they maintain shape of the cell. Cytoplasm is a translucent, homogeneous colloidal semifluid filling the space between plasma membrane and nucleus. Cell organelles are present in it. Functions help in manufacture and distribution of substances within the cell and in exchange of material between different cells of cell organelles. Third part is nucleus. Nucleus is small, located in or near the center of the cytoplasm, and network of chromatin present, chromosomes present in as chromatin. They are bounded by may be present on endoplasmic reticulum. They attach external wall of the endoplasmic reticulum. Functions of the endoplasmic reticulum is they gives rigidity to the cell and they help in the synthesis and transport of various proteins and fat within the cell to the outside. Second is ribosome. Ribosomes either scattered freely in the cytoplasm or attached to the endoplasmic reticulum, and ribosomes helps in for protein synthesis. They are the sites for protein synthesis. Next is mitochondria. Mitochondria is also known as powerhouse of the cell. They are minute sausage shape or rod shaped granular bodies scattered in the cytoplasm. Functions of the mitochondria is first they carry out cellular respiration, and that's why they are called powerhouse of the cell, because they get energy and store in them during respiration. Next part is Golgi bodies or Golgi complex or Golgi apparatus. Stacks of flattened sacs or small vesicles generally located near the nucleus, and similar structure is found in Golgi bodies are a similar structure which is in a plant cell known as dictyosomes. And function of the Golgi bodies is they help in the secretion and storage of the substances like enzymes and hormones. Lysosomes are the small vesicles or sacs that contains digestive enzymes which destroys and digests the worn out cell parts. The function of the lysosomes is they clean up the cell debris and they help to rapidly destroy and digest damaged cell and their part. So that is why these are also known as suicidal bags. We have talked about nucleus, membrane nucleus, endoplasmic reticulum, plasma membrane, lysosomes, mitochondria, and there are some other parts which are also found in cell, that is vacuoles. They are non-living part of the cell, and granules too. So first we will talk about vacuoles and then granules. Vacuoles are fluid filled membrane bound spaces, large size vacuoles in a plant and smaller and fewer ones in animal cell. Vacuoles helps in storage of water and other substances. In case of granules, they are small particles, crystal or droplets, and granules function of the granules is they contains starch, fat that serve as food for the cell. So vacuoles are the space large in plants, as we have known, they store water and other substances. Protozoa have food vacuoles that digest food and contractile vacuoles remove excess water. In case of protozoa, example we can take amoeba and paramecium. Parts found in plant cells only we will talk about, that is cell wall and plastids. Cell wall, when we talk about cell wall, so plant and bacteria have the cell wall around their cell membrane made of peptidoglycan and cellulose respectively. Plant cell is made of cellulose and bacterial cell is made of cell membrane is made up of peptidoglycan, which provides rigidity to the cell and they protect. Cell wall is an outer rigid protective supportive and semi-transparent covering of a plant cell made up of cellulose. We have already discussed, and function of it, they provide a definite shape and rigidity to the cell, and they also protect the plasma membrane and internal structures or organelles. Second important part, plant part is plastids. They are only found in plant cells. Plastids are present in only plant cells. They have stacks of flat membranes forming grana interconnected by thylakoid membrane. And plastids are of three types: chloroplast, chromoplast and nucleoplast. Chloroplasts are green. They possess photosynthesis pigment, synthetic pigment, chlorophyll and carotenoid. Chromoplast contain yellow, orange or red color pigment, and nucleoplasts are colorless plastids. When we talk about the plastids, chloroplast contains green color, helps in photosynthesis. When plant receives sunlight, carbon dioxide, minerals and water, in the presence of chlorophyll, they help to prepare their food. And chromoplast provide color to the flowers and fruits. Nucleoplasts help in the storage of food. Now we will talk about the plant part and cell organelles which are found only in animal cell, that is centrosome. Centrosome is a small body lying above the nucleus, consists of two small granules called centrioles, and function of the centrosome is they participate in cell division and help in spindle formation during cell division. We have already talked about protoplasm is a living substance of the cell. Nucleus and cytoplasm together form the protoplasm. Now we will talk about differences between an animal cell and the plant cell. First we will talk about shape and size. Larger in plant cell, larger in size and rectangular in shape. In case of animal cell, they are smaller in size and oval in shape. Cell wall is absent in animal cell, but cell wall is made up of cellulose or present in plant cell. Vacuoles are found large in a mature plant cell. Usually a single large central vacuole is present. In case of animal cell, vacuoles are mostly absent, or if present are smaller in size and scattered. Golgi bodies are diffuse in the plant cell and are called dictyosome. In case of animal cell, Golgi bodies are well developed and present near nucleus. Centrosome and centrioles are absent in plant cell and present in animal cell. Plastids are present in plant cell. There are three types: chloroplast, chromoplast and leucoplast. In case of animal cell, plastids are absent. Food in plant cell is in the form of starch and oil. Reserve food in animal cell is stored in the form of glycogen. So, we have discussed different types of cells, their various organelles and its functions. Now we will move the next section, that is cell division. Just like a cloth, we wear out with time, continuously use utensils and they become weak and crack. So what we do for these? We just replace or discard it or purchase new ones. Same can't be done by from the body, but body can rejuvenate themselves or itself. So do cells of the body wear out and need to be replaced? New cells are required not only for replacement of worn out cells, but also for repair of cuts and injuries, for growth and reproduction. So new cells are obtained through cell division, but how does a cell divide to give two new identical cells? That is cell division. So we will talk about cell division. We have two types of cell division we will discuss. First, mitosis and meiosis. In case of mitosis, a cell gives rise to two identical daughter cells. Mitosis is needed for growth and repair of worn out parts, and meiosis, cell division involved in production of sex cells which give rise to the egg in female and sperm in male. So we will take little bit break. After break we will discuss. This discussion will continue from cell division.
प्रकाशित करने राहों को आलोकित करने हम अपना दीपक स्वयं बने, हम अपना रास्ता स्वयं चुने। हम अपना दीपक स्वयं बने, हम अपना रास्ता स्वयं चुने। जीवन ये प्रकाशित करने राहों को आलोकित करने।
धन साधन समय की सीमा से विद्यालय जो मुक्त हो, धन साधन समय की सीमा से विद्यालय जो मुक्त हो।
कभी पढ़े हम कहीं पढ़े वे विषय की लोच से युक्त हो।
कभी पढ़े हम कहीं पढ़े वे विषय की लोच से युक्त हो।
मित्रों हम उठे और जागे,
मंजिल आने की ध्वनि सुने। हम अपना दीपक स्वयं बने, हम अपना रास्ता स्वयं चुने।
जीवन ये प्रकाशित करने राहों को आलोकित करने।
राष्ट्रीय मुक्त विद्यालय का है द्वार खुला सबकी खातिर। शिक्षा परीक्षा का अवसर एक बार ही क्यों मिलता फिर।
शिक्षा परीक्षा का अवसर एक बार ही क्यों मिलता फिर। दुनिया में इसके केंद्र बहुत,
कोई मन चाहा केंद्र चुने। हम अपना दीपक स्वयं बने, हम अपना रास्ता स्वयं चुने।
before taking a break, we have talked about what is cell, discovery of cell, different types of cells and cell organelles, difference between plant and cell and animal cells. Now we will talk about cell division. Cell division means production of new cells, and there are two types, mitosis and meiosis. Mitosis, cells give rise to identical daughter cells, and in case of meiosis, the meiosis cell division is involved in production of sex cells. So when we talk about the dividing cells or mitosis division, so mitosis is an equational division resulting in two cells similar and equal in all respect to each other and the parent cells. They are similar to the parent cells. Mitosis helps to replace worn out cells, repairs injured cell, helps in growth and development of young ones, and this mitosis divides in four phases: prophase, metaphase, anaphase and telophase. At prophase, chromosomes can be seen, and by the end of prophase, nuclear membrane disappears. So major event of both kind of cell divisions are largely similar in both animal cells, plant cells. So we will describe mitosis in an animal cell. So first, chromosomal material inside the nucleus condenses to form chromosomes, nuclear membrane disappears, and centrosome divides into two equal parts called centrioles, each of which migrates to opposite sides to orient the spindle form, which forms the cytoplasm. You can see B nucleus condenses to form chromosome, C nuclear membrane disappear, figure C, and a spindle fiber of appears between the centrioles. So each chromosome consists of two chromatids which are held by the centromere, and these chromosomes are arranged in the middle of equator of the spindle, that is figure C. Centrosome splits, chromatids of each chromosomes now have their own centromere, and these chromatids, now new terms, chromosomes separated from each other and subsequently move to the opposite poles of the spindle, that is figure number D. Chromosomes lose their identity and turn into a network of chromatin threads at the two poles, that is figure E. Nuclear membrane reappears around each of the two new clusters of the chromatin material, form the poles. At the pole and the middle of the cell, at the two sides, a furrow appears in the cell membrane. The furrow deepens to divide the parent cell into two new identical daughter cells. So two main differences in meiosis in a plant and animal cell is, first is cytokinesis begins in plant animal cell begins with the formation of the contractile ring that pinches the cell membrane inwards, creating a cleavage furrow, and this furrow deepens until it completely divides the cell into two daughter nuclei. In case of plant cell, due to the rigid cell wall, plant cells form a cell plate. Vesicles containing cell wall nuclear gather in the middle of the cell and fuse to form the cell plate, which then grows outward to separate the two daughter cells and develop into a new cell pole. Second difference is centrioles and spindle apparatus. Animal cell centrioles are present and play a crucial role in the organizing of the spindle fibers that forms the spindle apparatus, and these centrioles move to opposite poles of the cell and are surrounded by estrus, which helps in spindle formation. Plant cells no centrioles, instead of the spindle fibers are formed from the polymerization of the tubulin monomers at microtubules organizing centers without the formation of estrus. So these are the two main differences. Now significance of mitosis. Daughter cells receive the same number of the chromosomes as the parent cell, and we can say another word, mitosis is an equational division in which the two daughter cells are identical to each other to their parents. Mitosis helps in wound healing and replacement of cells lost during wear and tear. It is also responsible for the growth of an organism by addition of new cells, and it is a method of asexual reproduction in single celled organisms like amoeba. Next type of division is meiosis. This process ensures genetic diversity in sexually reproducing organisms, and meiosis is necessary for sexual reproduction. And in case of animal, meiosis takes place in reproductive organs like testes and ovaries. Testes produces sperms and ovary produces eggs, and also in case of flowering plant, it occurs in the anther and ovary to produce pollen grains and ovule respectively. So now we will talk about the stages of meiosis. Meiosis is completed in two phases. First phase, phase 1, two cells with half the number of chromosomes in each are formed at the end phase 1. That is therefore a reduction division. And second phase or the second division is occasional in the mitosis and produces 4 cells at the end, each with half of the number of the chromosomes. So now we will talk about the sequence of event during meiosis. First, meiotic division. Chromatin fibers condense into chromosomes. Chromosomes are in matching pairs. A matching pairs means one chromosome having been received from the mother and the corresponding one received from the father. Both chromosomes have a pair, bear same genes but not necessarily the same alleles. So each chromosome in such a pair is made of 2 chromatids, as duplication of chromosomes occurs before cell division begins. So each pair of chromosomes is now a group of 4 chromatids. The nuclear membrane disappears. The homologous chromosomes which had pairs now begin to separate and move apart. Cytoplasm divides into 2 cells, each of one has the half number of the chromosomes originally present in the cell. So each chromosome is still made up of 2 chromatids, as centromere has not divided. This is reduction division, and meiosis 2 is exactly like the mitosis. At the end of meiosis, 4 cells with half of the number of the chromosome. So we can say 4 haploid cells are formed, which ultimately become pollen grain, ovule, sperm or egg. So now we talk about the significance. What is the significance? During meiosis, number of the chromosomes is halved in the resulting sex cells, so that when a male cell and female cell combine during fertilization, the normal number of chromosomes in the species is restored, and also new combination of genes are obtained in the gametes that result from meiosis. So this is the comparison between mitosis and meiosis. First, purpose is for mitosis is for growth, repair and asexual reproduction. Purpose of the meiosis is for sexual reproduction, that is production of gametes like sperm and egg, anther and ovule. Number of division in mitosis is 1 cell division, but in case of meiosis 2 successive cell division, that is meiosis 1 and meiosis 2. Number of daughter cells produces 2 identical genetically daughter cells. In case of meiosis, 4 genetically unique daughter cells are produced. Chromosome number diploid, or 1 number is of chromosome as parents, but in case of meiosis, haploid, half of the number of the chromosomes. Genetic variations, no variation found, clone of the parent cell. But in case of meiosis, high variation due to crossing over and independent assortment. Where did it happens? Mitosis is in somatic cells or body cells, but meiosis in germ cell, means testes and ovaries. And phases, when we talk about the phases, prophase, chromosome condense. In case of meiosis, homologous chromosome pair up and crossing over occurs. Metaphase, chromosomes align in the center in the mitotic division, but meiosis, homologous pairs align meiosis 1, then chromatids meiosis 2. Anaphase, sister chromatid separates in case of meiosis, homologous chromosomes separated, then chromatid in meiosis 2. Telophase, 2 nuclei form in case of meiosis, after telophase in meiosis 4 nuclei forms. So we have talked about what is cell, type of cell, the how, when we talk about the house runs smoothly when different members of the family and the helpers perform different household works. Similarly, different tissues perform different functions. So various tissues of an organism in coordination with each other in order to perform different processes that occurs in the body. So tissue can be defined as a group of cells similar in size, shape, performing the same function and having a common origin. So plants are able to produce new tissues throughout the life, but in case of animals can only replace some tissues under certain conditions. And example, we can take example, muscles of the heart and nervous tissue can never be formed again if damaged. So now we talk about the types of the plant tissues. There are two types, meristematic tissue and permanent tissues. First we will talk about the meristematic tissue. They found in the growing points of the plant, such as at the tips of the root, stem and branches. In figure 1, when we talk about figure 1, longitudinal section of the stem tip showing apical meristem, and is a meristematic tissue. Its breakdown of the two major growth center in plant, shoot apex and root apex. Shoot apex function, they drive upward growth and forms leaf, stem and flowers. Key part, the young leaves that initiate photosynthesis and protect the shoot tip. Pith, central tissue that store the transport nutrient, and root apex functions drive downward growth and forms root for water and nutrient absorption. In case of root cap, protects delicate meristem as a root pushes through the soil. Root apical meristem, that is the region of the active cell division. Primary meristem includes protoderm, ground meristem and root hairs. And why it matters? These apophyses are like the command center for plant growth. Shoot apex ensure the plant reaches light, while the root apex anchor it and gathers resources. Together they orchestrate the plant development and survival. So there are some main characteristics of the meristematic tissues. They aggregate of living cells compactly arranged without intercellular spaces. They are thin-walled and may be rounded, oval, polygonal and or rectangular in shape. Cells are small and have a large nucleus. They are capable of dividing indefinitely and add new cells to the plants, and they are usually found in the apophyses of root and shoot. Next we will talk about the permanent tissues. They are made up of cells which have lost their ability to multiply. Three types of permanent tissues are there: protective tissues, supportive tissue and conductive tissue. Protective tissues is made up of cells with thick walls and occurs on the surface of leaves, stem and roots. Supporting tissue provides support to various parts of the plants. This tissue includes cells that filled up the interior of potatoes, which is stored food found in the leaf stalks too. Conductive tissue is also known as vascular tissue, and it provides passages for the fluid to move up and down in the plant, and it is of two types, xylem and phloem. Xylem is located more towards the center of the stem. It allows water and minerals absorbed from the soil to travel upward in the plant. Phloem serves as to conduct the food synthesized in the leaves to flow downward and upward, so that food reaches all other regions. Now we will talk about the animal tissue. Animal tissues are grouped under 4 main categories: epithelial, connective, muscular and nervous tissues. Epithelial tissues are thin protective layer of the cell. They generally located on the outer surface of the body, on the surface of the internal organs and the lining of the body cavities. There are 3 distinct types of epithelial tissues: epithelial tissue, squamous, cuboidal and columnar epithelium. Squamous epithelium type, nature of the cells are hexagonal or irregular cells with thin walls. They are located cell of the outermost layer, and functions as the function of the squamous epithelium is protection of underlying parts of the body from injury, harmful substances and from drying up. Cuboidal epithelium cells are thick cuboidal cells. Location is some part of the kidney tubules and in glandular ducts, and the function of the cuboidal epithelium is secretion. Next type of tissue is epithelium tissue is columnar epithelium. Nature of cell is tall elongated cell. At some places have cilia at free ends. Location is inner lining of the stomach and the intestine, inner lining of trachea, and function is secretion, absorption, lashing movement of cilia pushes the material forward. Muscular tissues consist of long narrow cells called muscle fiber. They are muscle cells. They are so named because of their long fiber like shape. Muscles bring about movement of the body parts and locomotion in organisms. There are 3 types: striated muscles, unstriated muscles and cardiac muscles. Striated muscles, their contraction is under one control, so called voluntary. Nature of the cells is multinucleated cell, show bundles of the light and dark bands. They found muscles of arms, legs, face and neck. Function causes movement that are under the control of our will. Unstripped or unstriated muscles, also called smooth muscles, and they are leg transverse striation. Movements are under our will and hence called involuntary muscles. Slender typical tapering cells, they are this is nature, and location wall of blood vessels, urinary bladder, uterus. Muscles of alimentary canals contract to push that show peristalsis or food down. Function movement of the parts or content of the part not under the control of our will. Cardiac muscles, also known as heart muscles, exclusively present in the heart. They contract and relax rapidly, rhythmically and tirelessly, contracting and relaxing endlessly from early embryonic stage until death. Strip scenes on muscle fiber, short and branched, joined by intercalated disc. This is the nature of the cells. Location is heart muscles and function is contract and relax on their own. Connective tissue, as name suggested, connects organs. Basically, these tissues have matrix, connective tissue, cells and connective tissue fibers. Examples are areolar tissue, adipose tissue, cartilage, bone and blood. So, function of the connective tissues are they bind different structure with one another. Tendons bind bone to the muscle, ligament connect bones, forms a supporting framework. Adipose connective tissue helps in storage of fats, is also formed shock proof cushions around kidney, ovaries and eyeball. Blood is also in connective tissue. Next is fibrous tissue. Cells usually separated from one another by intercellular spaces. This space is filled with solid or liquid material. Example is tendon, ligament, adipose tissue. Function is connect to muscle to bone, connect two bones, packing and binding of most organs, store fat. Next type is cartilage, is a thick, semi-transparent and elastic in nature. In example or location is nose, ear, wall of windpipe and at the end of the long bones. They provide support and strength. Fluid connective next is Bone is a hard and porous matter, consists of both living and non-living and rigid mass of non-living cells. Location is rib, thigh bone, backbone. Function is provide support and strength, help in movement. Fluid connective tissue is contain cellular and fluid part is nature. They are example is blood and lymph. Function of connective tissue is transport of gases and chemical substances, protection from disease causing germs. Nervous tissue consists of nerve cells or neurons. A bundle of nerve fibers or axons of nerve cells forms nerve. Nerve cells is the structure and function unit of the nervous system, and it consists of three parts: body, cell body or cytone, dendron and dendrites, and axon. Cell body or cytone has a prominent nucleus and cytoplasm. Cell organelles like mitochondria, Golgi body are present in the cytoplasm. Thread-like structure like dendrons arise from the body. One of them is long and called axon. Axon may or may not be covered by myelin sheath or medullary sheath. Sheath is constructed at intervals which are known as node of Ranvier, and the space between axon ending of one nerve cell and cell body or cytone of the another nerve cell is the synapse. Now we will talk about stem cell technology. Stem cells are undifferentiated cell of our body which can be made to divide by mitosis and differentiate into specialized cell type and also more stem cells. Stem cell therapy can be used to treat blood cancer, test new drugs and provide cells for gene therapy. So, stem cells are undifferentiated cells in our body which have capacity to undergo mitosis and differentiate into specialized cell types and can redivide to produce more stem cells. Stem cells may be obtained from an embryo and umbilical cord and bone marrow in adults. Medical research shows that stem cell therapy can replace tissues damaged due to human disease. A number of adult stem cells therapies already exist, such as bone marrow transplant to treat blood cancer. Stem cells have potential uses to replace damaged tissue, to study human development, to test new drugs and to devise methods of gene therapy. So, this is all about what we have talked about, building blocks of life, cells and tissue. Now, we will recap in the form of main points. First, a cell is the structure and functional unit of the all living cells. Cell membrane is selectively permeable. It allows only selected substances to pass through it. Nucleus controls all metabolic and other activities of the cell, hence it is called the master of the cell. Endoplasmic reticulum help in intracellular transport, hence it is known as the circulatory system of the cell. Ribosomes help in protein synthesis, so they are called protein factories of the cell. Mitochondria are miniature biochemical factories which where food stuff are oxidized and released to energy is released, and this is energy is stored in the form of ATP. So, that is why it is called power of the cell. Tissues can be defined as a group of cells more or less alike in size, shape, performing the same function and having a common origin. Permanent tissues is a group of cells in which growth is either stopped completely or for the time being. In epithelial tissue, cells are closely placed and form a continuous sheet. The cell of the epithelium tissue rests to basement membrane. Muscular tissues consist of long narrow cells called muscle fiber which is held together by connective tissue. Blood and lymph are the connective tissue. Stem cells are biological cells which can divide through mitosis and differentiate into specialized cell types and can self-renew to produce more stem cells. So, this is all about cell and tissue. We will come again to meet a new program of science and technology. Till then, goodbye and thank you.